Saturday Morning Physics is sponsored by the M. Lois Tiffany Endowment, the Hideko Tomazawa Endowment, the Physics Department, and by gifts from friends of the program.
So the topics I'm going to cover today, more or less in this order, are what the role of the Museum of Zoology is, both on campus and internationally, what we look like, what our resources are like, how we serve as part of an international infrastructure for science, what we do, and how we teach. So what's the role? The first, most obvious role of a museum is that we archive and document biodiversity. We also provide resources for international research programs about the origins and maintenance of diversity, ecology, and environment. We send out internationally hundreds of loans every year of our specimens, and we receive specimens from other museums around the world, and I'm talking literally every continent and everything from places like the American Museum of Natural History to small colleges and universities. So we're servicing thousands of researchers. We also support internally a world-class faculty, research faculty, and their students, graduate students, and frankly a large number of undergraduates. And we teach at all levels, partly because we're also all faculty members in the Department of Ecology and Evolutionary Biology. We also traditionally have provided leadership in conserving biodiversity, and I'll give you some examples of that. What I really want to emphasize today is that the research collections in our museums comprise both the objects, those meaningful objects, and the information that goes along with those objects. So most of our specimens, in some divisions, nearly all of them are preserved in fluid. They were fixed in formaldehyde, and they're sitting there in ethyl alcohol, which is just basically vodka. And I know what you're thinking, and it's true. We also have dried specimens, skins, skulls, pinned insects, mollusk shells, so lots of, frankly, dead things. We also have tissues. We have large collections of tissues that we keep in preservative fluids or in ultra-cold freezers. We have lots of accessory information about those organisms, including movies of breeding behavior. We've got nests, we've got sound libraries, we've got photographs, we have x-rays. We have an enormous amount of accessory material. Although in the old days, when people used to ask us what a specimen looks like, we would just slap it on the Xerox machine and fax it to them, but we don't do that anymore. And we have all of the locality information in most of the collections in digitized form and accessible on the internet. Here's what we've got. I'm the curator of fishes. We have 3.5 million fishes down the street in 50,000 lots, bottles of fishes. I'll show you some in a minute, and 10,000 skeletons. And remember, all the data are available on the internet. In the insects, we have 5 million penned insects. And we have Barry O'Connor, who's our mite specialist, who's here today, has 300,000 slides of mites and about 2 million specimens. That's a rough guess. I don't know how he came up with that. 4 million mollusks, 250,000 catalog lots. Those are also, many of them now, online. That's actually a low count, 210,000 specimens of birds. Reptiles and amphibians, over 420,000 specimens, 13,000 skeletons, and about 175,000 mammals, and that runs all the way from tiny little shrews to a whale. So here's what the research collections look like. This is the fish collection. I'm really proud of this. This is the largest fish collection associated with the university in the world. And that brings me to the comparison. Our peer institutions in these kinds of research museums are Berkeley and Harvard. That's who we compete with for faculty and research grants. Again, as I said, these are in 70% alcohol, and every single bottle has a label in it, and all the information on that label is on the internet. We have 10,000 skeletons. There's a colony of domestic beetles in the museum, which we keep under tight control. We don't want to let them out. They'll eat the earth. And we put specimens in, and the larvae eat the flesh and give us the skeletons. We also have something called cleared and stained specimens, where we run the specimens through a series of enzymes and stains, and you end up with the skeleton in situ, so you can see the thing as it was in the fish. And I have actually done dissections of these things. The specimens are about 1 centimeter long. That's some tough dissection. We also have special preparations, and this is a CT scan of an African fish, and these allow us to do virtual dissections. This is all in computer, and we can go through the animal like this, and there's its brain case, for example. You'll see these bright spots here are the ear stones that are inside the skull that help the animal balance. And here, there's some great big ones, and then we run on out to the end of the skeleton. This is especially useful for very rare specimens. I'm working on, in fact, I just submitted a manuscript on a fish that became extinct in the 1880s. There's only 30 of them in the world. One of them is a completely disarticulated skeleton, and we just can't dissect these things. They don't make them anymore, so we can't clear and stain them. So we put them in the CT machine, we rebuild a skeleton in the computer, and that's the way we're able to do our virtual dissections. Oops. Don't want to leave the insects out. We've got 4.5 million of these things. These are some of the larger, showier ones. These are Barry's mites. There's a fur mite on the bottom right. He claims these are organisms. I can only trust that he's correct. He's got some that are so amazingly specialized that they actually live in the hollows of the quills of bird feathers. I mean, he's got an amazing. There's just amazing stuff that he tells. This is the mollusk collection, 250,000 of these. Traditionally, mollusks, they would take the mollusk shell, throw away the mollusk, keep the shells, so we have lots of shells, but in more recent years they keep the whole thing now, so they're able to do a lot more with it, and I'm going to come back to that later. Here's the bird collection, or pieces of it. We have everything from tiny, tiny hummingbirds to ostriches, harpy eagles. These are African sunbirds over here. Reptiles and amphibians, I should have gotten a cobra or something, but there's some bottles of snakes. As you can see, we have Bambi in our collection. I used to actually carry this specimen right here over to my comparative anatomy class under my arm, and as I was walking across campus, you could hear people weeping. Got skins and skulls. They also have mammals in alcohol, and they're really, really ugly. They also have a huge tissue collection, lots of skeletons, and bacula. A number of mammals have a bone in the penis, which is called a penis bone, or if you're a scientist, you call it a baculum. We've got lots of facilities in the building down the street, and I'll just quickly tell you what we've got. Aquarium rooms, live animal rooms, a venomous animal room where there's only one key and there's a special net to keep the critter from getting out of the room. We've got a vespiary. If you've seen lately the homepage at U of M, there's an article on the front page about the work that Liz Tibbetts does on wasps, and we supply her a place to keep her wasps up on our roof where we don't go. I mentioned the domestic beetle colony. We have ultracolds for archive storage. We have an X-ray soft X machine and a sound laboratory. This is the aquarium room. Well, I mentioned earlier that we have specimens and we have data. And the data for many people are actually, in a sense, more important than the specimens, although the specimens serve as the voucher that guarantees the accuracy of the data. But I want to talk just briefly about what the data look like. This is the catalog at the British Museum. These are specimens that Charles Darwin collected, and the standard procedure was to write them into ledgers, and sometimes you'd have a note area where you talk about the ecology of the area where the specimens were collected. And this is the way it was here at the University of Michigan, certainly in the fish collection. These are the fish catalog, the ledgers, and they're beautiful old leather-bound volumes with lines, and you can see the writing here. And you can also see that things are scratched out. So as names change and as concepts change, people go back and scratch them out and write in new names. And when we started our computer cataloging system, the system that we had was actually designed by us, and it made changing these names really easy to do. And we purposely designed a workaround so it was really hard to do, because we had two older curators in the department who disagreed about the taxonomy of North American fishes. And we could imagine one of them coming in the morning and doing a batch search and replace of a generic name, and the other guy coming in the afternoon and changing it back. So we decided maybe it would just be better to make it really difficult. One of them actually never learned to type, so we didn't need to do that. Those catalogs were transcribed into index cards, so we had 150,000 handwritten and typewritten index cards. All these cases were full of index cards, and then back in the 70s, we got a National Science Foundation grant to digitize those, so we hired people that actually keyboarded those 150,000 cards into the computer. When they did that up in herpetology in the late 60s, early 70s, the person that they hired to do it had to actually go away for a while. So the catalogs, all except for the insect division, the catalogs for all the divisions, at least some or all of those catalogs are online, and those databases are available to anybody, a wide range of scientists. The collections are networked, sometimes by organism-specific networks like Ornis is birds and Manus is mammals. One of the things that's happening internationally is an effort to get all of these data compiled in ways that we can look at new questions with the data, and I'll talk a little bit about that in a minute, the Global Diversity Information Network. The other information that we have that's associated with these specimens is field notebooks. When you're out in the field, you don't just, write a label or something. People write long descriptions of the ecology where they are, the ways the specimens were collected, what the color patterns were on the organisms, what the weather was like, what associated plant life was like, what the people were like, how they collected them. There's some places, there's a particular herpetologist I know, and if you look at his collecting localities from his field notebooks and look at the places where he was, it actually is places where there were places of alcoholic beverages being served. People go to fish markets, they talk about fishing procedures, and sometimes somebody will just write a little note about how they're feeling that day, and well, we went swimming and the food was good. And these field notebooks contain a wealth of information, and in fact, we've actually had ours subpoenaed in federal conservation lawsuits because they describe areas the way they looked in the 20s, the 30s, and they've been changed since then. We also have online databases that look like this. This is one of the bird databases where you see the specimen, you see the associated information, and again, all of that's available freely online. I just want to emphasize again that the museums house the real thing. We have the organisms, not just pictures, not just some artist's rendition, but the real thing. And that means that anybody, anytime, maybe 100 years, 200 years from now, unless the Higgs boson comes back and annihilates us, will be able to come to these same specimens and apply new technologies, ask new questions, and verify that the specimens are actually properly identified. Real briefly, because I want to, well, we'll just do this. The research in the Museum of Zoology is done by faculty curators who have half-time appointments in the Department of Ecology and Evolutionary Biology and half-time in the Museum of Zoology. We also have collection managers who are specialists in these groups and who help us coordinate and manage the collections. Every curator in the collection is expected to be a world's authority on the group of organisms that they are a curator of, and their research is in these kinds of areas. I'm not going to read all these because most of you, I'm pretty sure, can do that for yourselves. But I do want to point out that we study things from the molecular level to the macro level: behavior, color, DNA, complete genomes, coevolution, lots of a wide variety of evolutionary topics. And we also are studying things having to do with climate change, invasive species. And the Tree of Life project, which I mentioned right here, is one of those international cooperative programs that physicists are used to, where you have a handful up to 50 or 60 scientists working together using the scientific infrastructure to ask questions about, in this case, the phylogenetic history of life, the evolutionary history, which they are putting together in these major projects. So I want to give you a couple of examples of the kind of research we do, and I chose, well, frankly, my own graduate student, because I think what he's doing is really cool. It's Jeremy Wright, who came here originally to work on the systematics of a group of African catfishes. And being exposed to the kind of environment we have here, his research interest blossomed, and now he's working with another faculty member, Tom Duda, who's a specialist on venoms. And Jeremy's actually looking at the molecular evolution of venoms in catfishes. And he's doing it at every level that I just talked about. He's describing new species. He's looking at the histology of the venom glands and the spines. He's trying to characterize the proteins that make up the venoms. He's looked into behavior around what's eating catfishes, what's not eating catfishes, and why or why not. And eventually he wants to sequence the genes that are actually producing the venoms. Serendipitously, as a part of his research program, he noticed that there were some very distinctive color patterns in some of the African catfishes he was looking at, and he determined that this is a case of Mullerian mimicry. Mullerian mimicry is a situation where we have a series of closely related organisms with very distinctive color patterns or some signaling device that tells a predator, here I am. They're called aposematic color patterns. And if a predator has a bad experience with one of them, they associate that color pattern or behavior or whatever, this aposematic signaling. That's not a good idea, let's not do that again. And so presumably that then protects the other members of the group that has that color pattern from a predator. It assumes that the predator can actually remember the color pattern. And what's interesting is that there are only a couple of cases of vertebrate Mullerian mimicry. It's common in insects. You've all heard about the mimicry complex in insects. But in vertebrates, there's only two cases known. One is a pair of coral snakes, and the other is a series of frogs. And Jeremy has discovered a third one. And that has to do with the Tanganyikan catfishes in the genus Cynodontis. It involves a very distinctive color pattern. And there it is. And there are 9 species in this group, all of which have this very distinctive color pattern. And he's done a whole series of tests, and I can't go into all of them because it's just too complicated. But here's an aquarium with a barrier, and sometimes the barriers are impermeable, sometimes they're just netting. He's trying to eliminate the possibility that it's smell, and there's a whole series of experiments that he did, but I'm going to show you what this looks like. So he used a local predator, a bass, naive predator, and there's the catfish down here, and the bass is hungry. He made sure it was hungry. And the bass really, 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 really wants that catfish. Well, here's where the bass got the catfish, and it spits the catfish out, and it's clearly not happy from having bitten that catfish. Whoa. Okay, 60 days later, there's the catfish, there's the bass. Yeah, there's no question that that bass has had it with that catfish. Now, he's also done things like this where he's presented the catfish with mimics, or models made out of styrofoam and painted, and sure enough, if you show that bass this mimic, that bass will do the same. It sits on the far side of the tank. Show it a regular local catfish without that color pattern, and that catfish is gone. The bass eats the catfish. And show the bass something that Jeremy said, well, that looks kind of like a fish. Well, the bass thinks it's a fish, and the bass was so excited about that little model at the bottom that it actually broke through the barrier and attacked it and tried to eat it. And Jeremy had to kind of patch it. You can see the patches. So as long as he had those bass in the lab and confronted them with that catfish, they were remembering. They knew. So this is pretty good evidence that color pattern is quite distinctive. And I'll show you what that looks like. Let's try it this way. You can see that distinctive color pattern. There's the tail and the bars and the fins. And you'll also notice that there's a piece pulled out of this fish. And the piece is pulled out not by a bass, but by Jeremy, because now that tissue is in an ultra cold freezer, and it's going to be used as a part of his molecular studies. So that's one of several meaningful objects that you're going to see today. So Jeremy's work, which starts out as a systematic study of interrelationships of catfishes, turns into, serendipitously, this discovery of this third Mullerian mimicry complex in vertebrates, and the phylogenetic history. Here's his catfish with all the different color patterns, his phylogenetic history, which shows that this color pattern originated at the beginning of this group and has been carried along through its entire evolution, and they've been mimicking each other for a long, long time. Let me change the subject now to climate change. Phil Myers, who's curator of mammals, has been studying the distribution of small mammal faunas in Michigan for some years now. And it's turned out that it's been really crucial research about climate change in Michigan. What he originally noticed, he teaches a class up at the University of Michigan Biological Station, and as part of the class, they go out and they collect mice. They all learn how to collect mice. And he noticed that the mice that were supposed to be there, according to the records that they had, weren't there. And he got really interested about that and decided he better take a look into it. And so, of course, the first thing he does is he goes to the collection, he looks at the records, he looks at the field notebooks, he looks at the specimens, and that's important because the mice that he's looking at are very similar, and you have to be sure they're properly identified, so he did that. And what he discovered is that mice from the south are moving north into Michigan, and the Michigan mice that used to be here are moving further north up into Canada. And that's interesting for a number of reasons, one of which is that the cooler microhabitat mouse was the reservoir for hantavirus, and the warmer mouse that's moving into Michigan is a reservoir for Lyme disease. So you get to choose your disease by deciding where you want to be, but clearly public health people need to know that this is happening. Here's another example that he looked at, and that's flying squirrels. I don't know how well you can see it, but there are two different squirrels. Here's the southern squirrel and a northern squirrel, and here's in the late 1880s to 1980, you see that here most of the flying squirrels are down here, and now, 81 to 2006, they've moved up here. And the northern flying squirrels, which were already in the north, have moved way up north and now are virtually not found in in lower Michigan. And this is the case for all of the small mammal biota. They are responding on a much faster rate to climate change than, say, the vegetation is. I mean, trees have much longer lifespans. These small mammals have short lifespans, and they're responding to changes in climate very rapidly. And you can track it because we have all this information, we have the specimens, and we have the data, and you can basically do a time series from the 1880s right to the current time. Possums and climate change. This is another mammal that's used to be fairly uncommon in Michigan. You can see back in the 1880s to 1980, they're down here. You've probably all seen them waddling through Ann Arbor, getting into garbage cans. Notice that the records we have now to 2007, and here's probably some of you have already noticed that they're tracking the highways as people drive up to their cabins up north. This is I-75, and here's 23. There's Route 2. So one thing that all collectors do is take advantage of what's available, and if roadkill is what it is, you get roadkill. And that's exactly, and so these possums are now up north going through your garbage. And I think probably never before in Saturday morning physics has anybody kind of brought out a dead possum. So I thought I'd do that as kind of a beginning. So here's a possum, and I'll invite everybody up at the end of the lecture to come up and if you want to touch him, you can. It's a teaching specimen. And a lot of the things I'm going to bring out here, I'll let you folks come down and take a closer look, but I just couldn't pass that one up. Another thing that we're able to do with the kinds of information we have is to track the possibility of what happens when invasive species comes in. Invasive species are a larger and larger problem because of international travel, and as we all know, the Great Lakes is just full of exotics. And one of the things that we're able to do with the kind of data we have in our collections is to predict what the distributions of invasive species might be should they get access to our ecosystems. This is a snakehead. This is not the one that they found in Washington. That was a different species. This is one that is naturally distributed in the southern parts of Asia. But if it were to be released in the United States, it could be very happy, or in North America, it could be very happy in any of these environments. So there's a whole project called Life Mapper that uses, like SETI, they use people's computer cycles on their home computers to search through the databases around the world, get the distribution information, and then make predictions about what could happen if these species got distributed in other environments. Okay. Let's talk a little bit about our efforts in conservation, and here again we have some significant, meaningful objects. Many museum-based scientists, biodiversity scientists, we got into this because we love critters. And when you love critters, you hate to see them being damaged or destroyed or becoming extinct. So a lot of us get involved in conservation and to try to, keep our critters. And so we are personally involved, and we also use the available information that we have to try to aid in conservation efforts. Here's an example that's a very good one. A former curator in the Division of Fishes, Bob Miller, was an expert on these little killifishes, desert fishes. These are fishes that were left over after the great ocean that covered the western U.S. dried up. There were little pockets of fresh water left behind, and there's these little killifishes scattered all through the desert. And this particular one, Cyprinodon diabolus, lives in basically a sinkhole, and the entire habitat of that fish, it lives on a shelf which is covered with algae, and they eat the algae, and the whole shelf that they live on is about the size of this desk. You can see the entire species you can stand on the shore and see the entire species. And back in the 60s and 70s, because of agricultural interests pulling groundwater up out of the area, the water started sinking in their little tiny pond. Lawsuits ensued. It went all the way to the Supreme Court, and the Supreme Court ruled for the fish. It's a different era. That lawsuit and the organizations that came about because of that lawsuit eventually ended up in the Endangered Species Act. So, yeah, we can take some credit here at the University of Michigan for being a part of that very important law. And I have a question mark about success because water is still dropping, partly now because of climate change. There's just less rain now. So if the water drops another foot, that species will become extinct. Except in somebody's aquarium, probably. Here's one where we failed. This is a little top minnow. It's a live-bearing fish called Gambusia. This was in a spring in Texas called the Good Enough Spring, but apparently it wasn't good enough because the Army Corps of Engineers and the folks in Texas, and I'm a Texan American, so I can say these things. The folks in Texas decided what they really needed there was a lake where they could go bass fishing and water skiing. So the Army Corps of Engineers built a dam, and the result of that was the extinction of Gambusia amistadensis. Well, it just so happens that Gambusia amistadensis, we have something called the holotype. A holotype is the specimen that's the official name bearer. A holotype is the unique specimen that you look at if you want to make sure that your identification of a specimen is correct. There it is. It's about that big. It's unique, and they don't make them anymore. Why didn't some zoologists collect them and keep them alive in aquariums? And the fact is, they did. And as often happens, the best laid plans, some unnamed people accidentally mixed another species in with the specimens of Gambusia amistadensis, hybridization took place, and the gene pool was completely obliterated. That's what happened. And in fact, it probably wouldn't have mattered anyway because the number of specimens available was probably not enough to keep genetic diversity there to keep that species going for any length of time. When Europeans first came to the United States or to North America, they confronted an ecosystem very different from the one we know now. There were forest bison in the forests. There were Carolina parakeets flying through the trees, even in Michigan, parakeets. There were passenger pigeons, which are large dove-like birds and here they are. These are the Audubon folio paintings. The passenger pigeons were so numerous that their migrations would actually blot out the sun for days at a time. There were millions and millions of those birds. The Carolina parakeet became extinct in the earliest part of the 20th century, I think, probably because of diseases they picked up in poultry farms and because they ate fruit, so farmers shot them. Passenger pigeons had the unfortunate characteristic of tasting really good, and so they were shot into extinction. And their breeding requirements included enough density, population density, that they all apparently felt comfortable enough to breed. And once those populations dropped too low, they were no longer able to do that. So I just went up to the bird division, and I thought I would show you some of these. This is a Carolina parakeet. And it's a beautiful thing. I guess, well, it won't work for that. You can come up and look at it afterwards. These lived in Michigan, these beautiful tropical-looking birds. And this specimen is, well, it's about 120 years old. I'll put that there. Here's another meaningful object. This is a passenger pigeon. And this passenger pigeon was, well, it was shot, obviously, in 1886. And the person that shot that bird shot it out of a flock of 25 birds, and that was the last flock he ever saw. So that was right as the time they were on their way to extinction. Here's actually a case of some research that was done in the Museum of Zoology, which is a little more optimistic. And this is partly to show you that we don't just all hang around Ann Arbor all the time. Some of us get to go off in nice places to do our research in the Society Islands, and I put this up mostly just in case you didn't know where the Society Islands were. They're a hot spot. They're sitting over a hot spot on the Pacific Plate, and as it's moving, about every 3 million years, you get an island that then sinks. And that fauna, the fauna of those islands, island hop, as an island comes up and one's sinking, they just move over to that one and evolve over there. Well, they're beautiful. These are the endemic land snails of the Society Islands. Most of them were endemic. Endemic means that that's the only place they're found. And they were in micro habitats, little valleys and mountaintops. And in the 1970s, Jack Birch, who is a retired curator of mollusks, had the arduous duty of spending several months in Tahiti, where he collected lots of these little partulid mollusks, and he sent them back to the museum alive. You probably couldn't do that now because they've got much more restrictive laws about what can go into airplanes. And they took the critters and they ground them up and they stuck them in a -20 freezer, and he was working on a project. His co-author unfortunately died before the project was finished, and they just stayed in the freezer. Well, in the 1970s and 80s, some brilliant soul decided that what Tahiti really needed was a giant African snail because they're tasty. Well, of course, this is him or her, or a lot of these are hermaphrodites, so you don't know. And of course, the first thing it did was start eating everything in sight. So they decided, well, gosh, we got to get something to eat the African snails. So they brought in another snail, which is a predator, which ate the African snails, but it ate all the partulids as well. So people realized this was happening. SWAT teams went in and grabbed up as many little partulids as they could and moved them into zoos. And it turns out that they went from 61 species to 5 in less than 20 years. And this is all sort of focusy. So this international program got together. They put them in zoos all around the world, hoping that they would be able to save that fauna. But then the question is, how much of the fauna did we get, and what are the possibilities of actually restoring the fauna? And this is where Jack and Taewon Lee and Dermot O'Wheel in our mollusk division got together. They pulled out those tissues that Jack had collected back in the 70s before there was any problem, and reconstructed using mitochondrial DNA, mitochondrial clade groups of these things to see how much of the diversity actually had been destroyed and how much conserved. And what they discovered was that all the major groups had actually been conserved in one way or another. And now this kind of information is going into the planning of where do you put them back if you can put them back? How do you try to restore some semblance of the original biota? And in fact, they even came up with the rather radical in some circles idea of actually putting these things onto some of the other islands to save the species by moving them to a different place. And they figured, well, they do this anyway every 3 million years. We just have a few less years to do it in. So it looks like this is a case where a museum collection made long ago turns out to be extremely important in a conservation situation that was never dreamed of when they were originally collected. Well, I'm going to show you a series of other meaningful objects just because they're really cool. This is a deep sea fish called gonostoma. And gonostoma is among a group of deep sea fishes which are hermaphrodites. They're in the deep sea, the population densities aren't very high. So it's hard to find a mate. And sometimes, you know, if you can't get a date, you know, the world must be peopled. So this is Gonostoma. This is a sequential hermaphrodite. These things have ovotestes. They have both. They got the whole deal. And I hope you can see this. Can you see? There are along the. Well, maybe you can't. Yeah, you can see those little dots. Those are the light organs. They have luminescent bacteria that they grow in these special cells that help them to do a number of things, including countershading. If you're down below one and you're a predator and you're looking up, they have a way to keep the light coming out of those light organs at the same luminosity as the sunlight coming down through the ocean. And they also have patterns that tell their conspecifics, well, I'm one of your gang, and by the way, let's try something besides being a hermaphrodite. So they can act as male or female, or if they're really desperate, they'll take care of the whole thing themselves. Kangaroos, I'm sorry all these are fish examples, but fish are just really cool. And I actually know something about them, and I could have spent an hour talking about Barry's mites, but fish I know something about. Kangaroos are South American catfishes that are actually parasites. And they live in the substrate, down in the sand or down in the silt on the bottom of the river, and they sort of sit there waiting. And when another large fish comes along, like one of these giant catfishes, which can weigh up to 300 pounds, this thing will come down, settle on the bottom, and the kangaroos come up out of the substrate and will find their way, as this one is right here, towards the gill chamber. Here's the gill chamber of that animal. And it'll follow the flow of water. They're called rheophilic. They follow the water flow into the gill chamber, and they'll attach themselves to the gills and bite the gills so that the gill starts to bleed, and then they suck the blood. And we have a series of pictures here. Here's the thing going into or getting ready to go into the catfish. Here we are affixed to the gills, and this is the specimen after it's done, and you can see that it's just jam-packed full of blood. So it's like a big mosquito. It'll let go of the gill, passively go right back out of the gill chamber, down into the substrate, and it'll sit there and digest its blood meal and wait for another catfish to come along. Well, that's pretty cool. But sometimes, oh yeah, I forgot about that. Here are the teeth that they use to attach themselves to the gills. They've got teeth on the outside of their mouths. They also have spines that they can, like most catfish, that they can lift up to affix themselves in there, to help lodge themselves in there. And of course, they have the regular mandibular teeth as well. So I said they go in the catfish gills, but sometimes they make mistakes. And I've got to thank Ray Troll is a wonderful artist who does some really wonderful biological stuff. And Ray did a series of these. He did the picture that I showed at the very beginning, and he's been kind enough to let me use these. And Paulo Patri is a Brazilian ichthyologist who was actually involved in what I'm about to tell you about and loaned me these slides. So sometimes it makes a mistake and it goes in the wrong place. And it can go in the back end or it can go in the front end. And what happens is that this generally occurs if someone urinates in the water. And the catfish is attracted to the urine stream and thinks, great, meal, and suddenly finds itself in somebody's urethra. Well, they don't like that. And so there are issues. This is actually, what I'm going to show you is actually the first documented cases, lots of written examples of this, but this is actually documented. Guy gets up in the morning, he's in the Amazon, he kind of wanders into the river, and he's standing a little deeper than knee deep, and he's taking his morning urination, and he just happens to look down just in time to see a kangaroo leap up the stream of urine and enter his urethra. And he reaches down and he grabs it by the tail, and it's really slippery. And he had to let go, and up it went. So he's thinking, this is a serious problem. I should probably do something about it. I think he probably just takes a leak back in the forest now. I mean, this is well known. People know you're not supposed to do that. He was just being lazy. Now, they also go the other end if you know. So let's see if we can do this guy. Okay, there he is. So here's my finger to show you something about the size of this. And you'll also notice that this animal has this dark streak in it. That's its last blood meal. So that guy was collected just after he ate. And you'll notice he's quite pointed at the front and broad at the back. And there's his head and his little bitty eye. So some objects are more meaningful than others, I suppose. Museums, research museums, are really about infrastructure for doing science. And I was over in England this summer, and I went to a conference on biodiversity informatics, and I just put up the logos of some of the organizations that were sponsoring that colloquium, and it turned out to be really, really interesting because there were people from over 40 countries there, from every continent except Antarctica, of course, all of whom were interested in finding ways to combine the resources that we have in our museums. One of those cooperative units is the Global Biodiversity Information Facility, GBIF, which has 300 providers, including all of our museum collections, and they have nearly 200 million data points. Now that'll be catalog information about organisms, it'll be localities, things like that. What we want to do as research museums is provide our data in this cooperative effort so that someday you could sit down at your desktop computer or your, I don't know, iPhone or something, and combine all of those things in that second paragraph. Biogeochemical information from the National Ecological Observatory Network, soils, climate, weather data from NOAA, vegetation, hydrology, population genetic data, genomic data, diet, physiology, all the kinds of things that we know about the organisms, and be able to not only answer questions about conservation and climate change, but questions that we haven't even thought about yet. And if there's anything we know about museum collections is that they are long-term sources of information that you can go back to over and over again with new technologies and new questions. And I want to kind of emphasize that by taking a look again at one of Phil's mice. This is a mouse that was collected in 1925 up in Elmira, I think, Michigan. And let's just take a look at what we can do with it. Here's the first datum about that species. It says it was described in 1909 in a revision of the genus Peromyscus. It's a Peromyscus. So there's the first piece of information. Subsequent collected specimens, you can look like this. There's Phil's mouse, and what I've tried to do, and this is only a small selection, of all the ways that that single specimen is interconnected with all kinds of other things. So we've got, that mouse came from that spot in Michigan. So here's Google Earth showing that spot in Michigan. Here's one of Phil's field photos of that spot in Michigan. Here's that datum as a part of the Encyclopedia of Life, which is a huge online effort to catalog all the living things on the planet. so that anybody from anywhere in the world with net access can go and see distributions and biology and breeding behavior and all of that for every organism that we've got. This mouse is also in GBIF, so it's one of those 300 million data points. We can go to that specimen, we can get sequence data for whatever gene we might be interested in because we've got tissue on that mouse. We can do, this is Dr. Tucker, Perla Tucker, who's a curator of mammals in the museum, is doing population genetics in hybrid zones in Europe. In this case, you can do population genetics of mice in Michigan. That mouse figured into the mammals of the Great Lakes region. So there's a handbook, there's a popular outreach thing that this mouse contributed to. The mouse skull, mammal specimens are usually kept as a skin and a skull. The skull itself is, just look at it. It's a three-dimensional object jammed with biological information. There's tooth wear information. There's information about diet, growth. You can analyze it chemically to find out what temperatures it lived in. You can use the old-fashioned caliper kinds of analyses of these things, or you can use modern morphometric methods that use deformation grids to look at development or changes among species. And this mouse also is in the animal diversity web, which I'm going to come back to in a minute. It's one of our major outreach tools in the Museum of Zoology. So there you have, one specimen and all this web of things that this specimen is involved in. And here he is, collected in 1925, before there were any computers, before there was GBIF, before there was concern about climate change. Here's the critter, and we can do all those things with that single little specimen. So I'm going to put him right there. Well, so I mentioned the animal diversity web, and I'm going to briefly talk a little bit about what we do in the museum's community for teaching. All of the museum faculty sponsor undergraduates and graduate students in their research programs, usually focusing on a particular group of organisms. I walked into the bird division yesterday to pick up the Carolina parakeet, and there were two graduate students sitting there putting bird shot into the skulls of I don't even know what kind of birds they were, measuring brain capacity in those birds. So we have all kinds of research projects going on. We also teach in the Department of Ecology and Evolutionary Biology, and the curators in the museum, there's 5 of us, I guess, now, see about 1,100 students a year, and at least 600 students a year come into the museum. We have classrooms in the museum, and they're exposed to those specimens. Here's Phil Myers, our mouse guy, teaching the mammalogy course there on the left. And imagine how it affects a student to come into a collection, and they're not just reading something in a book. They're not reading about Peromyscus and climate change in Michigan. They're actually picking up the mouse. They're holding the mouse in their hands. And that's just got to change their perception of what's going on in the natural world. In the other slide is a picture of a group of people huddled around a gorilla. A couple years ago, this very large male gorilla at the Cincinnati Zoo went in for I hate to say this, it's going to not sound good, went in for a root canal and died under anesthesia. Bad health insurance, I just don't know. So you don't waste a gorilla if you got a gorilla. So they called Phil up and they said, want a gorilla? Well, of course, it took a millisecond to say, of course. So he drove down to the Cincinnati Zoo in his pickup, and they loaded this frozen 300-pound gorilla in the back of his pickup, and all of us were just praying, please speed, because we couldn't wait to see him pulled over by a highway patrolman to explain that gorilla in the back seat. So he brings it to the museum, they get a forklift, they take it up to the mammal division, they stick it up in the classroom, and they start making phone calls to the medical school, to kinesiology, to anthropology, to EEB, and people from all over the campus descended on this gorilla and took it apart. And took it apart for their research, and they were faculty members, they were graduate students, they were undergraduates, and all the pieces went to the people who were interested in those parts of that animal for their research. And there's a wonderful picture that I love. There's an undergraduate who's dissecting the forearm of that gorilla as a part of her education at the University of Michigan. Well, she's never, ever going to forget that. You know, that's the kind of experience that we can provide students that very few universities in this country can provide. I just happen to have the gorilla. So here he is. And if you come down afterwards, I'll welcome you to touch him, but don't move him around because he's falling apart. The Animal Diversity Web is our major outreach tool at the Museum of Zoology, and it's, again, something that Phil Myers did with the School of Education. It gets 500,000 visitors a month. More important than that is that there have been over 2,500 undergraduates from 30 institutions that have contributed information to that project. The Sweetland Writing Center, in fact, right now is working with EOL, Encyclopedia of Life, and the Animal Diversity Web to institute a program to have our undergraduates learn better how to write scientific papers by contributing to this project. And many of our classes, our undergraduate biology classes, certainly my fish biology class, for example, part of the course involves their writing scientific articles for this website. And it's used by K through 12 and people all around the world. It hits 24 hours a day. And there are actually high school and middle school curricula built around access to this website. We also have a website which we invite you to visit where you can see our databases and some other things. All of the publications that have ever been published through our series of occasional papers and miscellaneous publications are all online for free. If you want to take a look at those, you're welcome to do so. And finally, what I want to do is say that practically everything that I've said about the museum as infrastructure, the Museum of Zoology, applies to the herbarium and the other research museums on this campus. That we are really valuable international resources that increase and enhance the reputation of the University of Michigan. We are ourselves research scholars, and we support research internationally. In addition to that, the museums are a venue, not just an exciting, but an inspiring venue for the training of undergraduate and graduate students. It's it's hard to come into one of these collections and not be changed somehow when you see these specimens themselves, can touch them, and can actually investigate them in some detail. So with that, I will thank you for your attention and welcome you down.
Saturday Morning Physics is sponsored by the M. Lois Tiffany Endowment, the Hideko Tomazawa Endowment, the Physics Department, and by gifts from friends of the program.